Bonjour à tous. Je vais faire cet exposé en anglais. And it's a pleasure to present this research project that is jointly co-developed with Fabien GANDON and Youssouf Tagzouti, who are present with us today. I will start with the very basics of biology. You might have heard of the central dogma in biological processes, and everything flows from the DNA to the metabolic level that corresponds to the phenotypic, the one that is the closest to the phenotypic level. So if you want to understand a system, there is a need to study all these different molecular levels. And the science that are aiming at studying those levels are called omics science. They are trying to see all those molecules. So my domain of expertise is for metabolites, and we will discuss, it's fine. And we will discuss this level in particular. The first message I wanted to present you today is that we are studying those different levels using two different techniques. The first is next generation sequencing, where we can sequence the DNA or the RNA, and a mass spectrometry that allows for the detection of ions, small molecules that are ionized. Those are extremely powerful techniques. We can measure thousands of proteins, thousands of metabolites, and so on. And the process is very much presented here. We would have a biological sample, so it could be virtually any living organism or environmental sample. This material will be extracted and analyzed on the mass spectrometer. And afterward, we have to process the signal and generate some interpretation. However, for a long time, there was a major bottleneck in the annotation stage. So annotation is how do we convert a spectral signal into a clinical information. So it would be a structural property, a part of the molecule as presented earlier, or sometimes the entire molecule. However, unlike in proteomics, so here we have proteins, where the sequence of the peptides are relatively redundant because we have a set of 20 amino acids or so, depending on the living system. When we are studying small molecules, the diversity of chemical properties is gigantic. So for small molecules, this is very problematic. And for a long time, the community has been struggling in interpreting the signal and converting this into a biological information. And of course, if we want to understand the system we are studying, this was the largest limitation. In the recent years, I would say in particular in the last 5 years, the community through interdisciplinary research was able to develop approaches that is partially overcoming this bottleneck. And so there is a missing oh. Yes, so one of my contributions in the frame of such an interdisciplinary project was to generate some of so it was the largest public data set at the time of reference spectra from compounds that were isolated by chemists for many decades. And this was the most diverse, the most abundant. And my colleagues in Germany, the Professor Bocker, they saw this release and they said, okay, now we have a critical mass and enough representative spectra to have some ambitious deep neural network project to predict, to better predict the annotation task. And so here it's just summarizing the different methods that Professor Bocker and Professor Dornstein have been co-developed. So it was the work I did as a postdoc. Prior, I joined the community of the University Côte d'Azur about a year and a half. And those different tools are allowing to resolve the molecular structure, some molecular properties, and even classifying the compounds into chemical classes. This was remarkable for many applications, because instead of being limited to a very small subset of information, we could really expand and perform a better analysis. After my time in California, I've joined the laboratory of Professor Jean-Luc Wolfender in Geneva, and there they started to study the largest plant collection in the world. So it's from the Pierre Fabre laboratory, a French pharmaceutical company. And this is an exceptional resource. However, studying sample at that scale has many challenges at the experimental level. First, to collect the data with the mass spectrometer and so on. So that was interesting. And at the same time, we are generating a massive data set, very heterogeneous, with plants from all around the world. And of course, how can we integrate this ensemble of information from the experimental acquisition to the data interpretation and so on? So for that reason, the lab there developed a method using knowledge graph that allows this entire representation of the experimental setup and also the signal that is generated. And the power of knowledge graph is that we can translate some question into data mining requests using a programmatic language. Here, this is Sparkle, the most commonly used one. And that question can be translated to a request. This request can extract information from the knowledge graph, and the expert can try to interpret the meaning of this. However, we had a huge discrepancy between the people that can formulate the magical questions and the people who could program or could code the request. And that was slowing down this process. So this happened at the same time when ChatGPT was released. And we thought, okay, maybe we can experiment with such large language model and see if we can convert this natural language question into a programmatic request to accelerate this process, this dialogue. And our early experiment at the time showed that, yes, with enough context and enough information, the large language model was capable of generating some basic requests. So we started to dream about this and even imagine that we would have eventually in the future a system that could do the entire flow from the question to generation of the request down to the answer, to the sorry, no, we don't see it, to the answer for the interpretation. Okay. So soon after, I've joined the CNRS here at the Université Côte d'Azur, and one of my first emails arriving on the campus was to contact Fabien GANDON, because there was an obvious, knowledge graph was new to me. I'm a chemist by training. And so the potential of knowledge graph was something very promising for chemistry, but you really needed the expertise of the semantic web and other approaches to really empower this. And we have welcomed this student from the MIT, Emma Tissinger, with who we started this project of the KGBot. So it was initially a prototype. And this system was designed to achieve this entire flow from the question to the answer. And to achieve this, we have established a multi-agent system. So I know agent would have a different meaning, but here, each agent that is symbolized by a symbol is a large language model that has an ability to take a decision regarding what to do with the information and which tool to programmatically access in order to collect, for example, the information needed for knowledge graph, so the entities, the schema, part the graph, and so on. So in the system, there are multiple agents that are interacting together to achieve this conversion from the natural language question down to the query, accessing the server, so the experimental data, and responding to the user. So we have a prototype that is working really well. This was co-developed with the Troisier à Côte d'Azur. And I will show you a few examples here. We have the web app with a question. So this question is very, c'est du jargon scientifique de chimiste. But essentially, this is saying, I'm interested in those molecules that are present in this plant species with those properties. Can you collect all the information from the 70,000 extracts you have available? And what we see is that the system, with some delay, because there are several LM calls ongoing and some cost associated to it, is able to perform some complex task like converting this question into a request and also providing an answer to the user. So it says, okay, here are the results. You can access this link and see the result by yourself. And also, the entire flow is indicated, and below we can see a part of the Sparkle query that was generated. Some other questions that are also, I will not translate this, but looking for molecules in plant. And what we are observing is that because the LLM has a limited context window, when there is a lot of information needed to answer to the question, then the system is saying, okay, this is the request, but I cannot run it. This is just too much information. So please take it offline and see by yourself. Okay. So, and as I said, we can see the different steps. So it's getting the entity ID, getting some information about the experiment, looking at the schema to assemble the Sparkle request, and so on. All right. So in this KGBot project that is supported by the Academy RISE, in this project, we have several specific aims, but essentially, we want to take this initial prototype that was designed for metabolomics and chemistry to a more sustainable architecture that is able to be generalized to other knowledge graphs. And one of the challenges that we are facing is that so far we use the OpenAI model, so the one used for ChatGPT. This is a commercial model, proprietary. Sometimes we see a lack of reproducibility as well, depending on the day, depending on the hour. So there are many issues with the initial impact. It was not designed to be highly modular. And so in this project, this would be essential to achieve this generalization capability. And secondly, we have seen in metabolomics that the user interface is critical for the adoption of novel tools in the field. So there will be a graphical user interface that is more advanced than what we have now, including the ability not just to respond with text, natural language, but also with visualization. For example, if my question is about, I want to see the trend in this, the enrichment in some chemical classes, there will be a dynamic process that would produce the needed visualization depending on the user question and the type of results that were obtained. And also, initially in the prototype, if we were asking the second question, the same question twice, it was doing the same process again and again. So there are several technical challenges into caching the information efficiently in order to be more efficient and to use less resources and to be faster, to respond faster to the user. Okay, and so last information, and this is relatively, this is very recent actually. So this project was the base of a larger proposal for an INR with the Swiss National Foundation project, with Fabien GANDON and other partners here and in Zurich, Geneva, Fribourg, and Lausanne, where we will be developing an AI assistant for metabolomics. This is the proof of concept in terms of we can do a multi-agent system for one application, but we want to have something that is an entire ecosystem that would bridge both prior knowledge using knowledge graph and experimental data, and that would be available for the OMIS community to facilitate the interpretation of data in this very interdisciplinary field where one needs to be an expert in mass spectrometry, in statistical analysis, and so on. So this is very powerful to be able to automate some of these tasks and so on. All right, and so Yusuf, we count on you in the support of the Academy RISE to implement this project in a larger consortium as well. So this is very exciting for all of us. And so I want to acknowledge and thank you so much for the support by the Academy, as well as the other funding agencies, so myself, CNRS, also funding from the INR, incoming funding from the INR as well, the Troisier Tech Pool that was very efficient in the early stage of the project, and the rest of the team, and all of you for your attention. Thank you very much.
Pas de questions?